Good morning, good afternoon, and good evening, everyone, and welcome to this Inside Scientific and DSI webinar titled Turning Innovative Features into Practical Benefits Within the Buxco Nose-Only Rodent Inhalation System. I'm Liam Sanyo from the events team here at Scientist.com, and I'll be your host today. We're being joined by Shemesh, who's the Senior Product Manager for Inhalation and Respiratory at Data Sciences International, and today he'll be presenting Buxco's Nose-Only Rodent Inhalation System and highlight how it can improve research efficiency and other valuable insights for inhalation respiratory studies. And with that, I'm very pleased to welcome Shemesh. Thanks so much for joining us today. And the floor is yours whenever you're ready.
Yeah, thank you, Liam, for the introduction, and thanks everyone for joining, either live or on demand. I appreciate it, and hope to bring some interesting content in the next hour or so. So let's dive in. Today we'll be discussing the nose-only inhalation system and research. Talk about the benefits and challenges really quickly. Just as an introductory purpose, and then we'll dive into a mock inhalation exposure study. We'll go through an initial setup, optimization, acquisition, exposure session. We'll look at some post-exposure analysis and alternative exposure methods. So just as a really brief introduction, I'm sure everybody on the call is within inhalation or looking to get into it. But when we talk about nose-only inhalation, there are certainly many benefits of choosing that route of delivery. You can expose multiple subjects in a controlled environment. In general, you get a homogeneous aerosol delivery with a relatively low amount of API, especially when you compare the whole body exposure route. Rebreathing is typically prevented, especially if you're using a flow-past system like this one and many others. And no dermal exposure, so there's no grooming and so forth, one of the main benefits. Also, scientifically, this has a low amount of side effects and a quick path to the bloodstream. So everything being equal, the inhalation delivery is a good choice. However, as we all know, there are also some challenges when you choose the inhalation route, or the nose-only inhalation route, I should say. The animals do have to be restrained. That does add stress to both the subject and the user. Chamber conditions are a big play. Flows and pressure management. This is a closed loop system, so this really matters. Relative humidity levels, whether they tend to be too high or too low. Many types of aerosol properties and the routes through the tower system, loss and other considerations, formulations and how they interact with the materials and so forth. It is considered to be a little bit more labor extensive and requires a certain level of expertise. So really the point of this webinar is to see how technology and how new features are able to take some of these challenges and mitigate them, make them a little bit easier for the user. We're all here in the inhalation community. We all want more and more inhalation research to happen, and we all want it to be as easy as possible. And I've kind of taken a new approach with this webinar. Instead of just going through slides, I decided to actually run an inhalation study, pretend it's like a day in the lab, and go through the software that's connected to the hardware, create a very simple inhalation study that you can see here. We have 4 subjects, 4 mice. We have a solution concentration of 15 mg per mil and a delivered dose target of 10 mg per kg. And we'll just go through the software and see how it's set up and how it's built and how we can make the most out of it with this system. So let's dive in. First, we'll do a study design and setup, which includes flow and pressure management, leak and regulation diagnostics, subject-specific dose targets, which will introduce us to smart study. That will be a big section. And we'll talk about nebulizer efficiency rate, or NEB-ER. So without further ado, let's jump into the software. When you launch the FindPoint software, you immediately get a very quick USB communication diagnostic. Basically tells you you are good to go. We're sending a few signals, and we're saying that the devices that are connected through USB are properly communicating. So nice and easy, just lets you move on. So we're good to go on that, and we are on to the first real page. And this is a nice page for us to configure the controller options. Here, instead of having the user decide what the flow rates should be and which sampling device should get what, we simply ask the user to enter their system configurations. So they put in the number of subjects, the flow rate that they want per subject, the tower pressure, and then what devices they actually have on the system. And then the software dictates all the flows by itself, and then saves it for the next time, so you don't have to do it again. So just a nice benefit of having the user not have to manage all the flow settings, which tends to be a bit of a quirky manner. Okay, so we move along. Once that is set, the next page offers the user an option to run a leak test. And a leak test is important because you don't want to run through an inhalation system only to find out afterwards, after the animals that were exposed, that the tube wasn't plugged in all the way, or something of the sort. So with one click, there's going to be an automatic test that tests the leaks, and then also tests that all the pressure regulation works properly. To be more specific, in the two green sections, we are going to test the actual tower connections, all the ports, the sampling ports, the tubing connections, as well as the control system itself. Again, we hate to find out after the animals are exposed that there was an issue that caused some sort of problem with the exposure itself. So we do that first. That takes about 45 seconds, and we move on to the next page, which gets to the heart of what we're trying to do here. Okay, so we mentioned that we have 4 subjects, and we mentioned that we have a delivered dose target. Now, something that's very unique to the system is that you can actually put in that target into the specific ID. So as we can see here, we have 4 subjects that are prepared, and they each have their names and their weight, and there's an AIA target. And AIA is going to be synonymous with delivered dose and gets us off track for the first time, as we need to talk about AIA and what that is. So AIA was introduced in DSI about 6 years ago, and has been a very popular feature. Basically, we are originally a respiratory company in Buxco, and we do that very well. And it was very natural for us to have respiratory parameters measured during the exposure itself. And as we were measuring every single breath, and we also knew what the concentration was, we were able to calculate how much aerosol the animal breathes in a single breath. And then when we accumulate that, we can actually get the real-time accumulated inhaled aerosol, again, the same as delivered dose, reported in real time, which gives the researcher a great insight into what's going on. Now we know that the animals, even if they're the same weight, they're going to breathe differently, right? They're going to have a different minute volume, different respiratory matters. There's many publications on that in the handout section, but that's not to be argued with. So now, with different breathing rates and different endpoints, we can actually have different delivered dose per subject, and we can actually report on that and measure on that. But the problem was that even if you have it, the different real-time delivered dose, or AIA measurements, the user didn't have an option of what to do with it if the subject achieved its target. You could pull it out of the tower, but that has its own concessions at that point. That ruins the test article environment, it's labor extensive, and there's atmosphere and lab contamination. So people didn't want to do that, understandably, which led us to an important feature that was recently released, which is called Smart Study. And let me try to explain what that is. So imagine again a four-site inhalation system, and you have a target for all of them, and they're all breathing at different rates, and the first animal achieves its target. Once the first animal achieves its target, it's understandable that all the other three have not yet gotten to that target. Again, they're breathing at different rates. Now, those are arbitrary numbers, obviously, but I believe that they're somewhat reasonable within the margins of error. So once the aerosol continues, the second subject achieves its target, but the first one is now over target. You go to the third and fourth, and now you have an ununiform exposure. So what we would really want to do is once the first subject achieves its target, we would want to close off that aerosol only to that subject, and then replace it with fresh air. Okay, so that's a technology that we've been wanting to do for a very long time, and now it's finally here. And let me give a couple of videos to see what is truly happening. Here we see the video on the left. We see the actual port when the animal restrainer gets attached to, and we can see that we're now able to close that jet where the aerosol comes out of. We're able to close it at the port. That technology is very important and allows us to actually control the animal's delivered dose. And you can see here on the middle video, it's a little bit faint, but you can see the aerosol coming out, and on the left port it just stopped, while aerosol continues to stream out of the port on the one on the right. And that is really the smart study ability that we were talking about. Now the one on the right just stopped and the left keeps going. And again, full control over every port. Okay, so we're back to here. And now that we're able to do that with each animal, we are able to get a uniform delivery specific to each subject. And that is Smart Study, and that is available now. And again, just to reiterate the point, once a target is reached, fresh air begins while exposure continues to the rest. All of that is done on a completely automatic setting. There's no user intervention that's required at all. And it's very, very important to note that while all of this is happening, the environment in the tower remains consistent. And we're able to do that by automatically reducing the flow, the system flow, and the nebulizer output to the same ratio that the ports close compared to the ones that remain open. So the concentration and the humidity levels in the tower will remain the same for the rest of the subjects that are still being exposed. Okay, so we're back. We're back into the software, and we know that we can specify a delivered dose or AIA target to the subject. But perhaps more convenient is to actually specify a mg per kg, because then the animal's weight is being considered, which is even more precise. So here we have our 10 mg per kg as a target. We now put in their weights. We can do the math and then put in the delivered dose with doing the math, but then you have to do that manually. Again, more convenient is just to put the weight into the software, and then the fine point will calculate that for you. So here's the weight, and then we've got the mg per kg. Okay, so we've gotten through setting up the system, checking it for leaks, configuring the flows and pressures, assigning IDs, and assigning their mg per kg target. So far, so good. Now we just need to set up the aerosol, and this is this feature of controlling the nebulizer is a feature I debated whether to put in or not because it's a very old feature. It's one of the original features of the system. But I still find that people appreciate that perhaps more than anything else. And the feature is, in fact, is that we can calibrate the aerogen nebulizer to a specific solution, specific intended solution. And by calibrating it, we know how much we can aerosolize in 1 minute under 100% duty cycle. Basically, the max we can get out of the nebulizer relative to that solution. And by knowing that, we can then specify a certain amount, a precise amount of aerosol to be aerosolized in a minute between the max and 0. And that gives you a very, very reproducible aerosol environment from one day to the next, one technician to the next, one study to the next. Very easy, very simple, and also as the nebulizer degrades, which they do tend to do, again, you can calibrate and then continue with the same precise aerosol output. So that really is a very convenient feature, and I just wanted to share some nice customer data that again look at this very tight distribution of the aerosol concentrations in the environment over the course of 6 different groups. Very low standard deviation, very, very good stuff. So let's move on. What have we done so far? We've set up the hardware. We've made sure it works properly. We've made sure we've assigned IDs. We've assigned them targets, and we've assigned an aerosol output. So we're done with the setup, but now comes the fun and perhaps more challenging part of inhalation, and that is the optimization of the system. And here we'll focus on aerosol characterization, estimated exposure duration, aerosol sampling correction, fixed concentration, and we'll actually run an acquisition and exposure session. So let's dive in. We mentioned that we can specify a ml per minute, an EBR. How much do we want to aerosolize in a minute? A very nice feature in the system. But how do you pick which value to put in? What is the implication of picking a certain value? That is one of the bigger challenges in inhalation, and sometimes people just turn it on full blast, and whatever happens, happens. Just maximize the dose and so forth without really understanding the implications. And what we tend to do is we try to give you the information upfront. So with this feature called aerosol characterization, what the software will automatically do is map out a relationship between nebulizer output and aerosol concentration and chamber relative humidity. And it will map that out across various spectrums. So here, just on the right, for example, we have a 0.09 ml per minute that is relatable to a condition that will be 24% humidity and around 500 milligrams per meter cubed concentration. So you know that if you pick that value, that's the atmosphere, that's the condition that you will be running at. So it really kind of trying to reduce the inhalation expertise requirement. You can do this on your own. It may take longer. This is done in a fraction of the time. It takes about 12 to 15 minutes to do it properly, which is still a very short time compared to perhaps hours or days of in vitro analysis. But more importantly, it just allows you to use the system with knowledge at hand. So you can change things accordingly. Let's, for example, try a new sample. We could just hit a new sample here, and then we can define the spectrum of aerosol outputs that we would like. These are fairly arbitrary here, but nevertheless, we run them, and then we get a new curve. And then again, let's just choose again. Let's suppose I want to run at 35% relative humidity, so I will choose the corresponding nebulizer output, which is 0.12 ml per minute, corresponding to 750 milligrams per meter cube concentration. And the nice thing here is that when I choose that NebER, Pfeiffer will also tell me how long the estimated exposure time will take, again, relating back to the exposure target, the mg per kg target that we entered earlier. Without that target, this would not be possible. Okay, this is all looks pretty good, but I'm sure some of you are wondering now, how do we really know that this concentration is accurate? I mean, we know that the photometer is, you can't really take that to the bank. So how much value does this really have? Well, that's why the next screen, the next option that comes to you in the software is the option to run a gravimetric collection. Now, running a gravimetric collection is certainly nothing new, and it's been done since the beginning of time. But what's unique here is that we can run the gravimetric collection and then enter its data into the software to correct the concentration and attribute that to the AIA, to the accumulated inhaled aerosol that's integrated with the respiratory parameters. Okay, so how do we do that? Very simple. We hit start, we run a collection. Obviously we have the filter. We're not going to show all that, but okay, we got a material weight. We enter it into the software. The software calculates how much the photometer is reading, and we get a correction factor of 0.826. Now, what do we do with that factor? That factor is automatically attributed back to the aerosol concentration, aerosol characterization that we ran earlier. So if you recall, before we were at 750 milligrams per meters cubed concentration, but now with the gravimetric correction, it adjusted it to 600. And now that will be used for AIA measurements. So we get a more accurate representation of the concentration. And if you're going to do chemical analysis, you can actually even take the vehicle out, so you're really only measuring the compound alone. Speaking of running compound alone, another very interesting feature that we recently added is the ability to put a fixed concentration that bypasses anything from the photometer. And in this case, you can put in a value that perhaps from the gravimetric collection, perhaps from a mathematical calculation of concentration versus output versus solution concentration. You can put in a bacteria count or anything that you think is appropriate to be integrated with their respiratory endpoints. And again, that bypasses what the photometer reads and goes right into the AIA. So both options are available, and as a matter of fact, this is my preferred choice, and we will use that. So we went with 600 milligrams per meters cubed. That was what we found out on the filter, and we'll put that into the fixed concentration value. Okay, so we optimized how we are going to run the system. We made it accurate. We made it. We created the proper environment, and we are finally ready to run. FinePoint, with all that data being in place, FinePoint can actually tell you how much solution you will need to achieve those targets. So here they tell us to put in around 2 ml. We will do that, and we are now ready to run an acquisition. So hopefully you could see that the optimization process that happens with inhalation all the time is made easier with some of these tools that are available within the software wizard. So let's dive into the actual acquisition. Once you are at that last stage, we're actually ready to run, which we are. So what we see here is the 4 subjects. We can see them all breathing. They're all through the mini vent, which is split into 4. So as you can see, the tidal volume is lower than a typical mouse. I'm sorry about that. But still, they're kind of breathing at different rates, and we can go ahead and start the aerosolization, and you can see the concentration jumping up as well as the AIA. So now animals are breathing in the aerosol. And we have the smart study window on the bottom. You can see the targets for each one of them at 10 mgs per kg. And you can see that they are climbing up. And you can see that they're climbing up at different rates. And they're climbing up at different rates because the subjects are breathing at a different tidal volumes, different minute volumes, which will happen with all subjects. We can modify the mgs per kg if we want, as long as we did not surpass that target. And we can also manually click the bypass if we wanted to close the jet for any reason. Perhaps we need to adjust an animal in the restrainer and so forth. That can manually be closed as well. What else do we see here? We do have an expression map, so you can decide what graphs you want to see, what parameters are more important for you in the dashboard, change the color and so forth, so you can very customizable to whatever is important to you. Okay, as we move along, we'll pass through and fast forward through this acquisition session, but we can see that the mouse number 4 has already reached its target, okay, and that's indicated by the green light there, and we can see that number 2 has just achieved it. And let's take a quick look at what happens when a subject achieves its target. Their concentration goes down to 0, only theirs. Only their concentration goes down to 0. The others have the same concentration. But also at the bottom, you can see that the system flow went down, and the nebulizar, the nebulizer output, also went down in proportion to the rest of the subjects, meaning that that is our way to keep the concentration the same throughout the experiment. Okay, so again, all of that is done automatically while pressure is maintained, environment is maintained, and so forth. So very, very hands-off type of system that targets the mgs per kg that we put out there. So as we move along into the 3rd and 4th, which should happen momentarily, we'll fast forward a little bit. And now the 3rd one has achieved it, and now we're about to get to the 4th one. When the 4th one, the last one, has achieved their target, that's the end of the session. And as you can see there, the measurement is placed, and the experiment concludes. So without user intervention, we've gotten 10 mg per kg to each of those subjects, taking into account their different breathing parameters. Let's save it, and then a report will be automatically generated, and this is an AIA report. And recall that since we put in different weights for the animals, they all have a different delivered dose. So we have one at 0.21 milligrams, second one at 0.2, third one is 0.2, and the last one at 0.22. So hopefully you found that useful, and we will move on to some post-exposure analysis. Okay, we will talk a little bit about in vivo imaging system correlation to AIA and respiratory endpoints, optimal animal flow range, AIA to PK correlation, and animal reduction potential. Let's dive in. A nice trend that I've seen with customers lately is actually taking an image of the animal and of the organs, and then looking back and seeing what was their AIA? Can I reproduce the same image by targeting an AIA, by targeting a delivered dose? And even more so, which I find very exciting, is to attribute it to a respiratory endpoint. At this point, in this case, it's a peak inspiratory flow. How does that correlate to an image? This is fairly new, not the imaging itself, but the correlation to AIA and respiratory. But I thought I'd share a nice application of how it's used. Something that is the next topic is optimal animal flow rates, and this is a fairly important one. I wasn't sure if to put it into the post analysis because it could really also be done in before in the system setup, but I find that too many users are using a static animal flow rate as dictated by industry standards. So a rat, a half liter per minute, a mouse, 0.3 or less. And then they don't necessarily change it because, and understandably so, because if you lower the flow rate, for example, there may not be enough fresh air for the animal, and we don't know how the animal is doing. But leveraging plethysmography, since we have the respiratory endpoints during exposure, we can actually find that range when the animal is breathing properly by doing a pre and post experiment. And I'm happy to see a couple of customers actually doing that, finding that optimal flow rate so they can have some sort of play with that flow. And how does that impact? Why would someone want to change that flow? Well, for example, if we look back to our study and we look at 100 mg per kg target for the subject, we can tell that we need 20 ml to achieve those targets. If we go back to that original screen where we configured the controller settings, we configured the flows, we put in 0.3 liters per minute. If we change that to 0.2, we would need 33% less material. Now, why is that? It's actually fairly simple to understand, right? Chamber concentration is defined as milligrams divided by liter. That means the lower the flow, the higher the concentration. Delivered dose is concentration times minute volume times duration. Duration is delivered dose divided by concentration times minute volume. That means the higher the concentration, shorter the duration. Shorter the duration means less API and less animal stress. Another way to sum up that data is to look at this chart that basically goes to this very standard inhalation setup, and seeing the three columns are 0.5 liters per minute, 0.4 liters per minute, and 0.3 liters per minute per subject. And if nothing else changes, you can see that there's potentially great savings on material, right? So I go back to this screen and I say, if you're able to assess that flow range, if you're able to see if the animal is in distress with less airflow, and when that happens, then you can feel comfortable to reduce that flow rate if needed. Okay, so that's a very a little bit of an advanced feature of the system, but it's there. At the same token, using that plethysmography data, we can actually assess a vehicle and API tolerability, right? So we can see if the subject is distressed by an increased minute volume or other respiratory happenings, and then that can give you a tool to change formulation again if needed. And we're moving on to I think probably the most important, I don't even want to say feature, the most important part of the system, and that is the correlation between AIA and PK. If you do run a PK analysis post-exposure, you can then correlate that PK to the delivered dose. That is a standard term for deposition fraction. Once that correlation is established, then the PK can be better targeted. Well, all of that is nothing new. But since we are measuring the delivered dose for each animal, a correlation can be made for each subject. And consider the customer data below, where actually they did not use smart study. They only used AIA. So they collected for a certain amount of time and then picked up AIA, which was actually different for all animals because they all breathe differently. But once they collected PK and you did PK divided by AIA, you can see that the correlation was the same. So now, if you had smart study, you can actually target PK. With that in mind, imagine a study that uses 12 animals, and 4 of them reached proper PK, the targeted amount of compound that was meant to be in the lung. Another 4 were off, but perhaps acceptable, and another 4 were just way off and discarded. You can do the same thing. We can achieve those same green 4 animals with smart study using only 4 animals, so we can target that PK. And that falls right in to one of the 3 R's, the reduction, any strategy that will result in fewer animals being used in research. Okay, so hopefully you can see that this is a tool, a technology, an innovation that fits that mold, that hopefully will get grants going, hopefully will get in the right direction, and I believe it's as good as any tool for that vision and for that strategy. All right, we're moving along, and we're off to alternative methods. Okay, so far I've mostly talked about plethysmography, about using plethysmography with inhalation. That was the main topic of the last session, but I want to stress that plethysmography is not required for this system or for smart study. And I wanted to point the various things we can do with a duration target. So instead of running smart study with a dose or AIA or mg per kg target, what you do need plethysmography for, you can say, you know what, I don't really need that precise dose. Maybe perhaps you're running a smoke exposure or something that does not require that really precise mg per kg. You just want to run different groups. You can do that. You can choose a duration target and put multiple groups on the same tower. Okay. So typically, let's suppose you had 3 groups A, B, and C, and you wanted to dose them for 1 hour, 2 hours, and 3 hours. That might be 3 different runs. And with 3 different runs, you got to clean in between and then reset up, and that takes time and so forth. Well, now with Smart Study, you can actually do that in a single run and simply have each group close the individual ports while continuing to aerosolize the others. And if you consider that it might take 1, 2, and 3 hours for the others, this one can only take 3 hours total. So pretty nice feature. Sure, you can do it in both ways, but time is money, so a nice efficiency there. And I wanted to bring up a very nice application that was recently brought up by a customer, and this is when the customer was trying to do a T0 necropsy, harvesting the organs, but could only do 1 animal at a time. So if they all came out at the same time, they'd have a bit of a problem because one would be harvested at T0 and another one would be harvested later. So what we did, we set up that they're all under the same duration but start at different times. And when an animal is removed, you don't actually have to open the entire tower, ruin the article concentration, get atmospheric contamination and so forth. The jet stops and the animal is removed, and you can see that they're staggered, in this case 5 minutes apart, but we're able to do the necropsy properly. So a very nice feature using the duration target. So with that, it's been a long presentation. Thanks for paying attention for that long. I'll leave you with a few takeaways. Typically, challenging nose-only inhalation setup and study design can be made easier by leveraging innovative technology, and software features. I hope you can see that in the study setup. Built-in study setups lead to reproducible, low standard deviation chamber environments. Go back to the nebulizer calibration, regulations of the flows and pressure, all of that done for you, click of a button, and you're off and running. Various tools exist to map inhalation conditions over various aerosolization output spectrums. Back to that aerosol characterization, get you that knowledge before so you can pick the proper environment. Correlation between delivered dose and response can be made per subject, again, taking account their unique respiratory endpoints, how much they're breathing in. They're all different, and let's treat them as such. Technology is available to take a step towards reducing animal count in inhalation research. We know it's going in that direction, so we might as well jump on board and do our best to make that happen. The ability to close a single aerosol jet has several benefits, but including multiple dose groups and improve user safety work environments using that duration target. So I hope you enjoyed this. I hope you were able to get something out of this. And again, I really do appreciate you taking the time and paying attention. Thanks, and I'll take any questions that people have.
Excellent. Thanks so much, UV. All right. Well, let's kick things off with a great question here. Somebody's written, I have a question about smart technology. How does that actually work? And specifically, where does the fresh air come from?
Yeah, good question. So each one of the smart study ports is going to be connected to a compressed air line. The compressor connects to the singular controller, and then from there, maybe you saw in some of the images that I showed, the red tubing. Each one of those red tubes signifies a connection to the controller, which is then connected to a clean medical air compressed line. That compressed line has two jobs. The one job is to close the jet, and at the same time as it closes the jet, it goes through a critical orifice within the port that then supplies that fresh air to the animal at the right flow rate. So it's just as simple as that. Not much else. The actual closing of the jet is done by a bladder that is again is compressed by the high pressure air. So good question.
Excellent, great answer. All right, next one. So thank you for the webinar, very informative. You seem to focus on pharma research a lot. Does the system also support other applications like smoke, gases, and other things like that?
Right. Yeah, great question. I do I guess I do somewhat apologize for focusing on pharma, which I did, but not I suppose didn't mean to leave out the rest of the applications, but it is true that I did focus on pharma. Many of the features that I went through do apply to various other applications that we support. Now, you mentioned smoke, so we do have a smoke machine and an e-cig vape apparatus that attaches to the nose-only system, as well as the other systems that we have: whole body exposure, whole body plethysmography, and we do gases, right, and many other applications. However, I should mention that not all the features apply to all the applications, which is why I really focused on the pharma side. So, for example, the regulation of the pressure and the flows are applicable to all applications. That doesn't really matter. The plethysmography and the finding the proper flow range, that applies to all applications within inhalation. However, the smart study portion of it, you do need the nebulizer, the aerogen nebulizer. And the reason for that is because we need to reduce the output of the nebulizer, when a smart study site engages. Again, if you recall that the slide where we maintain concentration levels, we maintain the chamber environment as bypasses smart study sites close, we need full control over that output. And since we don't have that perhaps over a smoke generator or a jet nebulizer, then we're unable to apply those features into those specific applications. So not meaning that, not to say that you can't use the system with it, but again, the smart study portions features are restricted to that, but all the other ones are applicable to all the other applications.
Excellent. Yeah, thanks. All right, how do respiratory endpoints get measured during exposure, and what kind of chamber is used, and doesn't that complicate the usage of the system?
Okay, right, big topic there. The well, first let's talk about the just the bare bones, basic, the technical aspect of things. How do we measure respiratory endpoints? I will refer you to the handout section, to a previous webinar, and also to a brochure to look at our plethysmography portion of the system. But basically, we're using what we like to recall as a head-out direct flow measurement plethysmography, which is the most common one, and fairly speaking, the most straightforward one, which is basically using a pressure transducer to measure the air, the pressure differential when the animal breathes in and out of the pneumatic. This is by far the simplest form of respiratory measurement when you compare it to, like, for example, to whole body plethysmography, which is indirect. So the measurement themselves are fairly simplistic as long as there is a sealed chamber. And I think that when you perhaps that second part of that question was, does it add complication to the system? Inherently, it does not. The calibration of the chamber is a single click calibration, and then you do everything the same, just add the back end plethysmograph to the restrainer. So in essence, it doesn't really. The complication perhaps comes with the data analysis. And I think that that's common whenever you add data points. There is more to analyze and more to look at. When you have sensors in a car, you have more data points, and okay, you have to pay attention to that. And again, we saw the various changes, the various ways that you can actually utilize that data for research benefits. I will just mention that I think the second part of that question was accuracy. Maybe I got that wrong, but if it was, there's a very simple way to see if the chamber is measuring accurately. The only way it does not measure accurately if there's a leak there, and if there's a leak there, there's a very simple way to see if there is, and that's if the flow signal is not centered around zero. That means that there's a flow going through the chamber which offsets the signal. So that's a very easy way to tell if it's accurate. I hope I answered the question properly.
Yeah, perfect. We do have an accuracy part here, and I think you sort of touched on it. Maybe you could elaborate a bit. How accurate is the ventilation measurement of the system, as it seems to be a pretty integral part of the system?
Yes, it is. Again, it's only as integral as far as the dose measurement, and this is something that you've heard me advocate in this webinar, perhaps in other platforms. If we are looking to get more accurate dosing, then we do need to measure the animal's ventilation, as it's different from one to another. So yes, the accuracy is important, and the accuracy, again, head out or direct flow plethysmography is the easiest one. And for us, I come from Buxco, 27 years in the business. The direct flow is the easiest one because it's a direct relationship between the amount of air that goes in and out of the pneumatic back to the volume. There is no fancy algorithms or anything like that. So the only thing you really need to make sure of is that there are no leaks and the calibration is proper. The calibration is as simple as it gets in our system, is the same as all of our systems, single click and you're good to go. If it's good, you'll get a green button, and if not, they'll let you know. Again, the only possibility for an accurate flow measurements and volume measurements is going to be a leak, and I guess I mentioned that in the previous question, how to observe that. But not very common. I would say that the ventilation accuracy is very good.
Excellent, great to hear. Um, could you elaborate a bit more on how we can assess the tolerability of new test compounds using plethysmography?
Right. Yes. So that was on the post-analysis slide. Uh, is again, we were happy to see customers doing that, and I presented some customer data that saw the minute volume of the animal increasing over time due to the what the customer believed was irritability to the buffer. And actually, a very nice way to do that is to actually take a singular smart study port and just give an animal, a control animal, only fresh air while the others are getting the API or a specific buffer that you're testing, and then see how the animal reacts to it from a respiratory endpoints. If it's distressed, you'll see that minute volume tend to go up as they're trying, as they're having more difficult time breathing and struggling. So that is certainly, it's not hard science, but certainly that data point is much better than having nothing.
Um, all right, next one here. We've been having issues with pressure management in our inhalation system. Can you speak on how this system achieved a slight negative pressure?
Our system, it's just a great question. So I take a little time here. Uh, our system handles it a little bit different. As a matter of fact, we've had a change in our own system 2 years ago, which I think was a change, an important change that may have been overlooked. What we do is we define the inflow, right, the positive air that goes into the system through that configurator that I showed in one of the earlier slides, where the user simply defines what they have on their system, number of animals, number of sampling ports, and so forth. And that inflow is set by the system, and it's static. That means that stays the same, which leaves us with the negative flow regulation. And we've been, and 2 years ago, we put the negative flow on a PID loop to regulate against a pressure target that the customer chooses. So if a user chooses any given pressure, the outflow, the negative flow will adjust itself in order to achieve that pressure. And what that does, the reason that it really helps is because there's not a lot of play when you choose a slight negative pressure, which is the most common pressure that people choose. There's not a lot of play since it's a closed system. So the tolerances in the mass flow meters can cause issues sometimes, and I don't know if that's the issue that you're having there. But the fact that we can move that outflow out in order to compensate for any tolerances really helps us to just regulate that pressure easily, where the user doesn't even see that's an issue. So again, the negative flow is on a PID loop. It's a dynamic flow that adjusts itself in order to meet the pressure target instead of previously it was static.
Excellent. Thanks a lot. All right. Do the delivered dose reports and respiratory endpoints provided by Buxco correlate well with the actual lung deposition confirmed through imaging techniques?
Um, that's a really good question, and I guess I would say that I was very happy to see our customers using that the imaging technique and relating it to dose and even to the respiratory parameters was a real surprise to me, and again, very happy to see that. But this is a fairly new trend, especially when you consider that it's related to dose, a smart study, and so forth, which is a relatively new feature. So I guess I would say that the data is just starting to come in. So I'm not comfortable saying that, hey, I'm guaranteeing you're going to see a correlation there for sure. I will say that the fact that the customers are using it and the data is starting to come in, that we should get some papers and publications and get the latest on it in the coming months. And I certainly hope for a fantastic correlation, and I think that hopefully we can update everybody that joined this webinar with those publications when they do come out. So hoping for good news. The data starting to come in, so I suppose more on that later.
Excellent. Yeah, I'll have my fingers crossed. Um, All right. So, what's this question here? What's the suitability for long-term studies, and especially to ensure the results stay consistent over extended periods?
Yeah, that's one of the bigger challenges in inhalation. To sometimes you have long longitudinal studies, and you do have to create the same conditions over and over again. And I think that the as far as suitability, I'll point to our most basic feature that maybe I overpassed too quickly during the session, but I really believe that the calibration of the nebulizer itself, and then the assignment of the amount of output per minute that the system can achieve uh, will yield reproducible results, consistent results from day to day. Because any nebulizer, is going to be a degrading piece of equipment, and the user can be none the wiser. So the fact that you can actually calibrate and see if it degrades, and then the software can compensate for that, allows to have a consistent output of compound. Add to it the regulation of the flows and pressures, which, again, without user intervention, there's no user error. And that, again, yields, a consistent run from one day to the next. And again, we can point to our slide, which we can go to here to see that the various groups over various days resulted in a very small standard deviation. Uh, so again, I attribute most of that. I'd like to say attribute to the nice fancy features of smart study and so forth, aerosol concentration. But I really believe that the suitability for long term, the best feature is in fact the regulation of the nebulizer flows and pressures.
Fantastic. All right, well, I think in the interest of time, we'll just have one last question here. UV, from a broader perspective, how do you see the future of inhalation studies evolving?
um, well, I thank you for asking me that. I guess I'm taken aback a little bit, but thanks for I appreciate the question, and for my opinion on that. Um, I really do believe that the inhalation industry is an old one, that has kind of been the same for a while, and I believe that there are benefits to be had from newer technology. Now, I understand that these things don't happen overnight. Far from it. Maybe it takes years. But I believe that in the interest of the three R's, that and moving from either reduction in animals or eliminating animal research, going to cells and so forth, I believe that there is a part for us to play, all of us, in trying to reduce the amount of animals. At least I don't think inhalation is ever going away. We're all in the inhalation community. Love to be in an inhalation community. Want to see more and more inhalation studies. But as we want to see more and more inhalation studies, I think we all have to be cognizant of how do we reduce the number of animals. That's the wave of the future, and that's where grants are. And I hope, and I'll do the best that I can to make to provide us with some technology that can lead us to that path. Again, I go back to the if you can achieve the exact same goal with half the amount of animals, then let's do it. So I guess that's my piece on where inhalation is going. Hopefully, again, I appreciate anybody taking that to the bank.
Excellent. Yeah, really fantastic answer. So yeah, UV, thanks so much for being with us today and sharing your insights. It's really been a pleasure having you with us.
It's been my pleasure. Thank you, everybody, for dialing in.
Yeah, definitely. Big thanks to the audience for participating. And last but not least, of course, we'd like to thank the sponsors, DSI and Harvard Bioscience. And in closing, we hope you enjoyed this Inside Scientific webinar, and we'll see you again next time. Have a great day, everyone.